Hello, this is Dr. Flügel. What follows is an introduction to our brand new lab on human physiology. We are all very excited and hope you will have as much fun as we had over the summer developing this lab. In this lab, you will be taking measurements on yourself to formulate hypotheses about various physiological parameters and then test these. The lab concept is similar to Lab 1, the MIT, and also to the Goldfish lab. Since like these two labs before, it has two parts. In the first part, you will be collecting data which will be aggregated into an online database, which is then used in the second part for t-test analysis. There are two presentations for this lab. One, to go over the data collection, which I will do here right now. The second part you should watch after you have collected the data. It will give instructions on how to use the aggregated database to its full potential. In this short 15-minute presentation, I will go over the following topics. Important for you immediately is on how to enter your demographic data. Please make sure to enter your demographic data before coming to lab. I will come back to this point later on. The concept covered in this lab will include how different physiological states relate to physical activities such as running, squatting, or exercise, or how these relate to physical properties such as height, weight, BMI, being an athlete or not. You will again propose a testable hypothesis based on the data collected, and we will also bring back the t-test to deepen your understanding in the interpretation of p-values. The laboratory skills you will learn in this lab include the use of a computer interface with various probes to collect data, how to take measurements on human test subjects using these probes, entering experimental data into an online database, how to research a topic related to human physiology, and furthermore, for this laboratory, you will write your third and last laboratory research paper to refine your writing skills. For this lab, we will continue to enforce standard lab safety. However, you do not have to bring a lab coat. Just remember to adhere to proper lab attire as explained in the lab safety video. Otherwise, you might miss your in-lab quiz or even a larger part of the lab. The overall theme of this lab is human physiology. Before we go any further, I want to introduce some of the physiological parameters you can explore in this lab, which are: how does heart rate relate to physiological conditions or physical activities? How does one analyze an EKG? How to analyze an EMG? How does one analyze lung volumes measurements? Some of this material you should already be familiar with from Alice 2. However, since we will be covering a wide range of topics related to human physiology, I will not cover these here in detail, but point out where to get some more detailed information so you can do some research for the ones you are more interested in. So, I'm going to go over shortly what measurements you can take and how the room will be set up. There will be 4 different stations you will rotate through, which are related to heart rate measurements, EKG, EMG measurements, grip strength measurements, and air volume and airflow measurements. You will be able to follow 11 different protocols during the duration of this lab using these measurement probes. Some of these protocols have more than one experiment. I will briefly go over these here now. This is a probe for Station A. Station A has a handheld wireless heart rate monitor. In general, the heart rate at which the heart beats is an important vital sign recorded and analyzed by physicians at the clinic and hospitals. You will measure the response of your heartbeat to various activities. These experiments explore questions like, what happens to my heart rate when I put my foot in an ice water bath, or when I exercise, or when I do squats, or when I cough? For our purposes, we can compare the properties of the heartbeat. For instance, there are differences in heart rate between different groups of individuals. For example, athletes versus non-athletes. Shown here are just a few matches from an internet search related to this topic. This is the probe for Station B. Station B has a probe that can either measure an EKG or an EMG. An electrocardiogram or EKG is a graphical recording of electrical events occurring within the heart. An EKG is used to determine heart function and health. Doctors can look at an EKG and diagnose disorders of the heart such as abnormal slowing, speeding, irregular rhythms, injury to muscle tissue, and death of muscle tissue. For our purposes, we will measure the different components of an EKG, and then we can relate these to each other and ask different questions. For example, it has been observed that some components of the EKG can be different in between men and women. You could research this and then formulate a hypothesis, and then test it using the database we created. An electromyogram or EMG is a graphical recording of electrical activity within muscles. EMG is used in the diagnosis of disorders affecting muscles and the neurons that innervate them. In this week's experiment, you will analyze electrical activity in the extensor muscles of the forearm under various conditions, for example, using a barbell. This is a probe used at Station C. Station C has a probe that can measure grip strength in either your hands or your fingers. There are two protocols related to this probe. Testing of hand grip strength is used by orthopedic surgeons and physical therapists to evaluate the extent of an injury and the progress of recovery. Furthermore, grip strength can also be used to diagnose neuromuscular problems. As a rule, in diagnostic, it is assumed that there is usually a 10% difference between the dominant and the non-dominant hand in strength. If it deviates from 10%, then something might be going on. This shows an internet search related to this topic, and I found some article challenging this 10% rule. I think this is worth exploring, so you could test this or formulate your own hypothesis based on the relative strength between digits of the hand, for example. Another experiment explores the aspects of fatigue related to continuous gripping versus repetitive gripping. Here are shown some results on an internet search related to muscle fatigue as an example. This is a probe for Station D. Station D explores the measurements of lung volumes, which provides a tool for understanding normal functioning of the lungs as well as diseased states. Various questions can be addressed like, what happens to my lungs when I hold my breath, or breathe rapidly, or exercise? And this is it for the introduction of the probes. As you can see, we will collect lots of different data in this lab, which can be used to explore many interesting questions. Now, I'm going to talk a little bit about how to prepare for this lab and what to expect. First, please fill out your demographic data from home. Since you are sharing one computer, it might be awkward to enter personal data. Please go to the final website and then log in. There will be a link to the Human Physiology info page, which I will refer to the info page, or your info page in short from here on. Now, on the info page, click on the link Start Your Demographic Info page, which will get you to your demographic entry page. You will be assigned an animal name and a passcode. This is to make sure to connect your demographic data to your experimental data while keeping your identity anonymous. The top entries are sliders. You can also click next to the slider button to make them move. After filling out the form, it will change color to green or yellow. If you're done with filling out the form, please click Submit. Once you're done, the exit button will appear if you have entered all the necessary information. If the exit button does not appear, look for labels colored in red. And a tip, if this site gets stuck, just click on the link on the info page to reassess this page. Reloading the browsers won't work. Another tip, if you are not sure about the dietary choices, there are two small links labeled 1 and 2, which will reveal these two information sheets, which might help you to determine the type of your personal diet. Once you exit, you end up at the data entry portal, which only is accessible at the lab during your section or via the link provided on your info page. When using the experiment buttons on the left, you can access and look through each of the experiment's data entry tables, which might help you to get familiar with the data entry procedure. Also, once populated, you can find your personal experimental data here. Once you have populated your demograph page, your animal name and passcode will be displayed on your info page. Furthermore, there will be two links which will grant access to two pages, which are holding your personal, demographic, and experimental data. Both are accessible via your info page, which you can access through the final sign-up page. You can make changes from home to your demographic data until experimental data is connected to it. Then it will be locked. And on this note, you will not be able to enter experimental data from home at all. This is only possible during your section time in lab on one of the laboratory computer stations. To facilitate the data recording and entry while using the probe software, you will receive one personal data entry sheet. Please fill in your animal name, section, and quarter on the top. This sheet also has the same layout as each page on the data entry website to facilitate the data transfer. This sheet will be collected by your TA at the end of section. Now, let's have a look at the room setup. There will be 2 stations per room for each of the probes. You will work in groups of 3 as usual. You will start on one of the stations, A, B, C, or D, and then rotate about every 20 or 30 minutes. Some stations may take more time than others, so you might not get to all the stations. Just know that not all of you have to be the test subject for all of the experiments. There's an extra station in the room, station 9 by the spec. You can use this station to enter your demographic data if you not have done so at home, or to enter experimental data. Now, let's look at what you will find at each station. Each station has a binder with instructions on how to perform the measurements. Each binder includes abbreviated materials to conduct each of the experiments, consisting of data collection instruction, data analysis instructions, and background information. All these documents are already posted and available to you on CCLE. The posted documents on CCLE, however, have more detailed information than the abbreviated documents in each station. This is an example of the data collection sheet, which you should read first, so you can get right to the experiment. You need to run a test or trial run for each of the experiments first, in order to get used to and familiar with the equipment. If an experiment fails, it's probably easy enough to just repeat it. The experimental runs are usually short. Each instruction comes with a graph on what to expect. It also will show the timeline for each experiment. The data analysis sheet instructs on how to process the values from the graphs to parameter values, which are then used in the data entry. Initially, you can jot these down on the data entry sheet, and subsequently, you will enter these into the online database in between experiments. The analysis procedure on your first station might present quite a learning curve. However, all subsequent stations are identical in terms of how to use the interface and software, so each station will go faster with the progression of the section. Once you have time and a free computer, you can enter the experimental data into the online form. Let's look at how this is done. You need your animal name and passcode. To get access to the data entry page, please go to the Human Physiology website directly and not from your info page on the final website. Then, select your animal name and enter your passcode. If you're in the right section at the right time, the Enter Demograph button will appear, which means you have now access. When you click on the experiment buttons on the left, you might get one more question. This is related to your coffee consumption of today. If you indicated that you drink coffee, then you will be redirected to your demographic data page. However, all your personal data will be hidden. Only the coffee choices will be visible. So please update as instructed on the page, otherwise you will not be able to enter data. Once you exit from the demograph page, you will now be presented with the following page. Please start transferring your parameter values from the datasheet and hit Submit. After data entry, the page will look like this. To return to the main menu, click on the Back to Menu button. Don't use the Back button on the browser, this won't work. Once you are done with one experiment and you exit, your password will be cleared. If you have to enter another experiment, just provide your passcode again. The Go to Demograph button will not appear this time, since after entering data, the demographic data will be locked down. Now, a word regarding the background information. In order to get through the data acquisition part, please read the background beforehand, or only if you have downtime in lab. Don't get stuck trying to understand the details of each experiment first. You do not need to know the details to get through the data collection part. Also, sometimes the background makes more sense once you have done the experiment. You can then think about what factors might be influencing the outcomes of these experiments. Again, all these documents are posted in more detail on CCLE. The background provided in the folders is just so to get you started. Once you are done with the experiments, you can start focusing on the ones which hold your more interest. And then you can start reading through the more detailed background, just for your experiment of choice. Then you can furthermore investigate with web and/or literature searches the topic you find most intriguing, and then formulate your hypothesis and to start writing your report. Here are some guidelines for your last laboratory report. Your TA will go over the specifics with you in your section. Please again, submit your report with cover letter as described in the online lecture titled Scientific Writing, which also includes general guidelines for each of the sections of the paper. With this, I will conclude this presentation and wish you an exciting week of investigation in our laboratories.